At this point, you've learned about Mendelian genetics and probably have looked at some classic Punnett squares. Now let's take some time to talk about some deviations from Mendelian genetics. Because the thing is, Mendel really lucked out when he was doing all of his experiments. So if we focused on the flower color of his pea plants, for an example, this was a trait that is characterized by one gene in particular. And on top of that, that gene has two alleles. The dominant purple flower allele and the recessive white flower allele. And on top of that, one of those alleles is completely dominant over the other. So if we crossed two heterozygous individuals, like we're seeing in our Punnett square here, you get a very predictable outcome of one homozygous dominant individual, two heterozygous individuals, and one recessive homozygous individual. And this would be probabilistically if these two individuals had many offspring. But this is not always the case. And you were probably already onto this. If you think about certain characteristics like eye color, we know that if you have a parent with brown eyes and a parent with blue eyes, you don't always get this characteristic. 3 out of 4 children will have brown eyes if it's dominant, and 1 kid will have the blue eyes. Instead, there's often more of a spectrum, so things don't always work out as perfectly as Mendel studied it. So there's a couple scenarios where things don't follow those Mendelian genetics. First, we have when alleles are not completely dominant or recessive. Second, when a particular gene has more than 2 alleles. So with the pea plant, we had purple allele and white allele, but what if there were many different variations? We have instances when a single gene can actually produce multiple phenotypes. So instead of just one gene being linked to one trait, like the gene for flower color and a phenotype of different flower colors, what if we have one gene that can influence many different things, say, in the human body? We're also going to talk about X-linked traits. So this is when we have genes, but instead of being on your autosomal chromosomes, it's found on the X chromosome, which is your sex chromosomes. And that can cause some kind of interesting inheritance patterns. And then finally, we'll mention polygenic traits. Okay, so there's a lot going on in this image here. On the left, you'll see that we have 2 flower colors for a snapdragon plant. The red coloration is homozygous, as is the white coloration. So there has to be 2 alleles of the red gene to have, there has to be, yes, 2 alleles on each chromosome for the red flower to show up, and we have to have 2 alleles of the white flower variation. If you take 2 of these individuals, right, these 2 homozygous individuals, and you crossed them, we are ensuring that each of the offspring would have at least 1 allele for red and 1 allele for white. It's a little blurry in this photo, I have to admit, but what you're noticing is that the offspring, the heterozygote, is not red and it's not a white flower either. Instead, it has this pink intermediate. This is called incomplete dominance. It's when we see this pattern of one is dominant, the red, and one is recessive, the white. However, if you were to do your Punnett square, instead of seeing the ratio we saw before of a clear dominance of purple flowers and white flowers for Mendel's case, here we're seeing an intermediate color, a pink. Now, if you take the 2 heterozygotes, that's what we're seeing on the right-hand side here, so 2 of our pink flowers, and you crossed them, now we get more of a variety. Remember that for every genotype that a plant has, let's say if we're focusing in on this flower color, the plant has 2 alleles, and it can only pass on one of those to its offspring. So for the first plant on the left here, actually for either of them, they're both heterozygous, there's a 50% chance the plant's going to pass on the red allele and a 50% chance it's going to pass on the white allele. So we have a 25% chance that the offspring will get 2 red alleles, a 50% chance that the offspring will also be heterozygous, and a 25% chance that the offspring will have the 2 homozygous white alleles and show a white flower type. So again, this is called incomplete dominance. And a hallmark is that the heterozygote condition is an intermediate between the 2. Now, incomplete dominance can sometimes get confused with codominance. In codominance, we see an interesting interplay between different alleles. However, both alleles are dominant. So think codominance, they're dominant together. Whereas in our other example with the pink flowers, it wasn't that the white allele was being expressed and the red one was also being expressed. Instead, that white flower coloration is actually a lack of pigment. And just having one of the red flower alleles simply wasn't enough to produce the red color, so we get this nice pink intermediate. Here, for blood type, there's a variety of things that can happen, but what I'm focusing on for codominance is actually this AB blood type. So for blood type, you have your red blood cells, and they have different glycoproteins on the outside of the cell being projected off of the surface. This is actually really important for cell signaling in your body. It's one way that your immune cells know which cells are yours and which cells are invaders. That's why if I were someone with blood type A, which I believe I have, and I was given a transfusion with blood type B, my body would see those B glycoproteins on the surface of those red blood cells coming into my body and say, like, you're not from here, you're an invader. So they're very important. And we have a couple different alleles here. So the big I is just the denotion for blood type, and usually the uppercase means dominant, right? So we have this I, and then there can be the A allele, there can be the B allele, there can be type O, which is actually 2 recessive alleles, so just like no glycoproteins. And then we can have A and B. So I'll explain this a little bit more in a second, but notice that A and B are both dominant, meaning that if I have A with a recessive allele, which is actually shown up top here, I'm still going to get type A blood. If I have the B allele with a recessive allele, I still get type B blood. But if I have an A allele and a B allele, that's when we see type AB. So it's codominance because both of the alleles are being expressed, and it creates a new phenotype instead of like some intermediate like we saw in the last example. Okay, but there is something else going on here, of course. So yes, we have codominance in the case of our AB blood type. However, we are also seeing an example of multiple alleles, making things kind of confusing. So thinking back to the beginning when I said that Mendel was really lucky when he was looking at flower color, like the purple and the white flowers, because there was really just 2 alleles happening there, the purple allele and the white flower allele. Here, you're seeing that there's the type A allele, which is dominant, the type B allele, which is also dominant, and on top of that, we have a recessive allele. Even just with 3 different alleles, we can see that there's a layer of complexity added, but in reality, for a lot of the genetic diversity in the human population, there's more than just 3 alleles. There can be many different types, so inheritance already just becomes really complicated. So this is kind of a funky diagram. You might want to even pause and just stare at it for a little bit, but what we're seeing here is that, let's say hypothetically, we have someone with type A blood, and this is AO, so it's actually the dominant A allele, and then they also have that recessive allele, is what this image is trying to say. And then similarly, let's say the other parent has type B blood, and they have one dominant B allele, and then they also carry a recessive allele. If they were to have kids, there's actually 4 different blood types that could be passed along, each with a 25% chance of showing up. They could have a kid that is type A blood and carrying one of the recessive alleles, a child that is AB blood, a child that is B blood, and there's also a chance that they'll have a child that is type O blood, meaning that child has both of the recessive alleles. So clearly you can see that this isn't following that 75%, 25% phenotypic variation that we see when we usually cross heterozygotes if it's a completely dominant allele and there's just 2 alleles. Okay, next up we have X-linked inheritance. And this is often looked at from the perspective of when there's a recessive trait that can cause certain disorders like color blindness. So for example, color blindness is X-linked and it's recessive. So here, let's say that we're looking at color blindness and you have one parent, the sperm-producing parent, that is unaffected. So remember that, we've got to remember sex chromosomes. Males have XY and females have XX. And this is X-linked, meaning it is a gene that is only found on the X chromosome. I also want to remind you that the sex chromosomes, X and Y, they're not homologous. If you have two X's, okay, you've got a homologous pair of chromosomes, but if you have an X and a Y, that's not homologous. They actually don't carry the same genes. So here we have the father is unaffected. The mother is a carrier. So in this situation, the color blindness trait is recessive. And I know from context because it says the mother is a carrier. If the trait was dominant, then the mother would be effective and not just carrying the trait. So some people are carriers, like I might be a carrier for the color blindness condition, but I just don't know because I have an X chromosome that's functional and masking that recessive trait. So if we were to do a Punnett square, which is kind of being shown here, and have all of our chromosomes separate independently, and we get one chromosome from the paternal line and one chromosome from the maternal line. Here's the possible combinations. So there's a chance that we can have an XX, so female, that is unaffected, an XX female, where they are a carrier. We can have an XY male, no color blindness, and then here we have the XY male who does have color blindness. So you're seeing from all the possible combinations, the male is the one that's affected. And that's because males only have one X chromosome, whereas females have two. So as a female, if a female inherits an X chromosome for color blindness, as long as they get one more X chromosome that doesn't have the color blindness trait, they're good. But if a male inherits that X chromosome with color blindness, they don't have another X chromosome to kind of rescue them, right, or to mask that trait. So they will have color blindness. And that's why I can think of a few of my friends who are colorblind. All of them are male. Actually, that I know of, I don't think I have a female friend that's colorblind. I wanted to throw this in here because it's quite interesting, but we can also have X-linked traits that are dominant. And you can see that, again, it's following, if we do a Punnett square, it's making sense, but it's not exactly what we would see if we had a completely dominant trait and just two alleles on a homologous pair. So on the left-hand side, we have a scenario where the mother is affected, but is a heterozygote. And in that case, half of the children are going to have the affected condition for whatever it is. On the right-hand side, we have a diagram where the affected individual is the father. They're not technically a heterozygote, right? Because they only have one X chromosome, so that's all they get. But if you were to do your Punnett square, you find that still only half of the children are going to be affected. But in this case, it shifts, and it would be only the females. So kind of interesting, right? How the Punnett squares, like, they still work out, the math is still mathing, but who is affected, which sex is affected, and how it gets passed on kind of varies depending on whether the gene, the alleles, well, the condition for that gene is dominant or recessive, and whether the mother or father, male or female, is a carrier or affected individual. So lots going on there. Okay. We also have, super interesting, something called pleiotropy. So this is when one gene influences two or more seemingly unrelated phenotypic traits. So again, so far we've just been talking about one gene and variations within the alleles. Here, this is a case where if an individual has, I believe they have to be homozygous recessive for cystic fibrosis, that genotype can affect many different phenotypes. So with cystic fibrosis, the gene impacted is the cystic fibrosis transmembrane conductance regulator. If you can think back to what you learned in intro bio on what goes on in the cell surface and membrane transport, this is a protein that sits in the cell membrane and helps transport primarily chloride ions in and out of the cell. Okay, now if you also remember about membrane transport and diffusion and osmosis, where solutes move, water follows via osmosis. So this membrane protein is really important for setting up osmotic gradients that allow for water to move in and out of cells and produce things like mucus. If it's not functioning, you end up with mucosal layers that are really thick and can clog up. So this is going to happen anywhere that membrane protein is needed, which, spoiler alert, is in many different organs in the human body. So it's important for all epithelial tissues, so lining the nasogastric tract, digestive tract, and even some other organs like the pancreas. So it's just one gene, but there's phenotypic impacts all over the human body. Okay, so those were all examples of variation at one locus, aka one gene. So what about when we have characteristics that are influenced by many genes? So we're looking at the opposite, right? I just gave you an example of one gene impacting many traits. How about when we have one trait impacted by many genes? One great example of this is eye color. That's the reason our little Punnett squares, they're not working out here. So eye color is influenced by many different genes, especially those that influence the production and distribution of pigments in the iris. So I have just a few examples here. OCA2 is involved in the production and distribution of melanin, which is an important pigment, particularly in the iris. The SILV gene is associated with the development and maturation of melanocytes. So these are the cells that produce melanin. And then there's a bunch of other ones, like for an example, the IRF4. This is an interferon regulatory factor. So interferon is a signaling molecule, and this gene codes for a protein that influences that signaling molecule. And I guess that's all to say that it's going to influence how eye color develops and how certain pigments are distributed. Another excellent example is something like height. So there's not discrete characteristics for height like short and tall, even though you can describe someone that way. Instead, if you got 20 people in a room and you looked at height, it's more continuous, right? So these polygenic traits are often quantitative and they exist on a scale of variation. So height in particular is determined by the interaction of 423 genes, and there's about 697 variants of those genes. So that almost speaks for itself, but something like height cannot be measured by just one gene. There's many different genes at play that are influencing that particular phenotype. So thank you for joining me on this kind of rapid crash course on deviations from Mendelian genetics.